Okay, welcome to Chapter 11, Immunity. All right, immunity is the protection against disease, and this is possible because of our immune system and its components. So many pathogens do not cause us harm because we have physical, chemical, and cellular defenses that prevent them from entering. If they do enter, these same defenses, namely white blood cells, will prevent them from spreading throughout the body. So epithelial cells in the airway passages prevent the entry of pathogens. Hydrochloric acid in the stomach kills many bacteria that have been ingested. Blood clotting is a mechanism that prevents blood loss and the entry of pathogens through open wounds. So the immune system is the action of lymphocytes in response to the entry of an antigen into the body. There are different types of lymphocytes that are capable of producing different types of antibodies. Antigens and antibodies are specific to each other. Lymphocytes will only secrete its antibodies if the proper antigen is encountered. So the immune response would be slow the first time an antigen is encountered. Sometimes it can take several weeks to build up enough antibodies to destroy the pathogen, which is why we might experience sickness for a while. If a person continues to encounter the same antigen, a response can be rapid, enough to entirely prevent symptoms at all. Okay, so the cells of the immune system are produced in our bone marrow. There are two types of cells that are involved in the defense of an organism, and they are visible when a blood smear is stained to show the nuclei. So phagocytes and lymphocytes are the two that we're going to be talking about, and phagocytes are really broken down into two, neutrophils and macrophages. Okay, so phagocytes are a type of white blood cell that is produced in the bone marrow. These cells are scavengers that remove dead cells as well as invasive microorganisms. Neutrophils are one type of phagocyte, and they form about 60% of the leukocytes in blood. They travel everywhere all over the body, and they often leave the blood by squeezing through walls of capillaries to patrol the tissues, and that's why you find phagocytes in the lymph fluid. So these cells are released in large numbers during an infection, but they do not live for a long period of time. After they kill and digest some pathogens, they often collect at a site of infection to form pus. Macrophages are also a type of phagocyte, but they are much larger than neutrophils, and so are found in organs like the spleen, lungs, liver, kidneys, and lymph nodes. So when these cells leave bone marrow, they travel in the bloodstream as monocytes, which will develop into macrophages once they settle in their organs. And once in the organs, the macrophages will remove any foreign matter found within them. And the cells live long. They play a really important role in initiating the immune response since they can't destroy pathogens completely. Instead, they cut up the pathogens to display antigens that can then be later recognized by lymphocytes. If a pathogen invades the body and causes an infection, some of the body cells that are under attack will release histamine. So this chemical, along with any other chemical that is released by the pathogen, will attract neutrophils to the site. The neutrophils will then destroy the pathogens via phagocytosis. So neutrophils are able to attack certain pathogens because they have receptor proteins on their surface that recognize antibody molecules and will attach to them. Okay, so here you see this model. So step one is chemotaxis and adherence to the phagocyte by the microbe, and then the ingestion of the microbe by the phagocyte. Okay, then formation of phagosome. Okay, fusion of the phagosome with a lysosome to form a phagolysosome. Digestion of ingested microbe by enzymes, formation of residual body containing the indigestible material, and then discharge of that waste material. Okay, so lymphocytes are much smaller than phagocytes and have a single nucleus that fills most of the cell. Both types of lymphocytes are produced in bone marrow before birth. B lymphocytes are white cells that remain in bone marrow until they mature. Once they mature, they spread out through the body, concentrating in lymph nodes and the spleen. These cells will release antibodies that destroy antigenic pathogens. And then there are T lymphocytes, or T cells. These are white cells that leave the bone marrow and collect in the thymus, where they go to mature. Some will coordinate the immune response, stimulating B cells to divide, and others seek out and kill any of the body's own cells that are infected with pathogens. So immune responses are only carried out by mature lymphocytes. During the maturation process, millions of lymphocytes develop. Each type is specialized to respond to one particular antigen, giving the immune system the ability to respond to almost any type of pathogen that may enter the body. Immune responses depend on the interaction of both types of lymphocytes to give an effective defense. Okay, so B lymphocytes. Each mature B cell will be capable of producing just one type of antibody. So during the maturation process, the genes that code for antibodies are changed in a variety of ways to be able to code for these different types of antibodies. Once each cell divides, those cells or clones will be able to make the same type of antibody. So at this stage, the antibody does not leave the B cell. Instead, it remains in the plasma membrane. The antibody then forms a protein receptor that will specifically combine with that one type of antigen. Okay. Some of the B lymphocytes will become plasma cells, which are then capable of producing antibodies very rapidly. And these cells secrete their antibodies into the blood, lymph, or into the linings of the lungs and the guts. And again, because these are the things that are coming into contact with these pathogens and are the fluids that are carrying these pathogens. So plasma cells do not live long. Their numbers decrease within weeks after the pathogens have been identified. However, the antibodies that they secrete into the bloodstream will remain for longer. Other B cells will become memory cells. And this is so cool. So these cells remain circulating in the body for a really long period of time. So if an antigen is reintroduced up to a few months after the first infection, the memory cells will divide really quickly and develop into plasma cells and more memory cells. And that reaction will occur every time there is antigen that is reintroduced into the body. And this destroys and removes the infection more quickly so that no symptoms of the disease even develop, which is super cool. So you don't even know that you've been infected because your body is fighting it off. Okay, so the primary response is slow because there are a few B cells that are specific to that antigen. And then the secondary response is much faster because there are now many memory cells which quickly divide and differentiate into those plasma cells. So there are many more antibodies produced during the secondary response, as you can see here. Right, and that totally makes sense. Okay, primary response is slow. There's only a few of those B cells. They need to find that specific antigen and then replicate. But because there are those memory cells that are hanging out in our blood for a longer period of time, our second exposure to that same antigen, the immune response is a lot quicker. Okay, so this brings us to immunological memory, and that's the ability of the immune system to respond quickly to antigens that it recognizes, okay, as already having been in the body. So memory cells are the basis for this and can last for years. But we can suffer from repeated infections because there may be many different strains, and new strains may have different antigens. Okay, so this brings us to antibodies. Antibodies are all globular glycoproteins and form the group of plasma proteins known as immunoglobulins. So all antibodies consist of 4 polypeptide chains, 2 long or heavy chains and 2 short or light chains. There are disulfide bridges that hold the chains together. Okay, each molecule has 2 identical antigen binding sites that are formed by both light and heavy chains. The sequence of the amino acids is what makes the specific 3D shape, which binds only to one type of antigen, right? So the hinge region provides the flexibility for the antibody to bind around the antigen. Okay, it's very important that you study this model and understand what each of these regions are, including the variable region on the heavy chain and the light chain. Okay. All right, so antibodies have different functions. Some of them act as labels, and they'll identify antigens as targets for destruction by phagocytes. Antitoxins are a special group of antibodies that will block the toxins that are released by the bacteria, which is interesting. And some antibodies that have multiple binding sites cause the agglutination of bacteria, which reduces the chances of those bacteria spreading. Okay, so here are the 4 classes of antibodies that you are required to know. Okay, so it's important to understand that I don't think you need to necessarily know the exact relative molecular mass, but you just need to understand which ones are smaller and which ones are larger. And you do need to know the number of antigen binding sites on each of these, the sites of action, and the functions. So you can see that there's some that specifically are working. I mean, most of them work in blood or tissue fluid. Okay, but you can see that there's some that are specific to like saliva and tears, or the bronchial secretions, okay, or tissues, etc. Okay, so this brings us to T lymphocytes. So remember, mature T lymphocytes have those T cell receptors that have a similar structure to antibodies and are each specific to one antigen. A T cell will become activated when they encounter the antigen. And this can occur after a macrophage engulfs and cuts it up and exposes the surface of the molecules of that pathogen. But it can also occur by a body cell that has been invaded by the pathogen and displays it on its plasma membrane. Okay, so there are 2 types of T cells that respond to pathogens. The helper cells, okay, the T helper cells, these release cytokines. Okay, cytokines are hormone-like chemicals that will stimulate the B cells to start dividing. That way they can develop into the plasma cells, secrete their corresponding antibodies, and really go on the defense. Other T helper cells secrete cytokines that stimulate macrophages to carry out phagocytosis more vigorously. So also there are killer T cells. These look for body cells that have who have already been invaded by pathogens. And that's because they're displaying those antigens on the plasma membrane, right? So that's how the killer T cells know. They attach themselves to the surface of the invaded cell, and they secrete toxic substances that kill the pathogen along with the host cell. So there are also the memory T cells that remain in the body and can become active quickly during the secondary response. Okay, active immunity is gained when an antigen enters the body and an immune response occurs where antibodies are produced by the plasma cells. So since the activation of lymphocytes occurs naturally during an infection, it is known as the natural active immunity. The immune response, however, can also be activated artificially, and this can be accomplished by injecting antigens into our bodies or taking them by the mouth, i.e. vaccinations. Okay, so this is known as artificial active immunity. The immune response caused by natural or artificial active immunity is similar. The antibody concentrations in the blood provide long-term immunity. So in both types, it takes a sufficient amount of active B and T cells to be produced to give an effective defense. So if a person becomes infected with a potentially fatal disease, then a more immediate defense will be needed. Okay, passive immunity. So passive immunity is gained without an immune response because the B and T cells have not been activated and plasma cells do not produce antibodies. Artificial passive immunity occurs when the antibodies come from another person who has encountered the antigen. So natural passive immunity occurs when antibodies are passed from mother to fetus via the placenta while pregnant, or from mother to child via breast milk. The antibodies that the mother produces can pass across the placenta or in the milk and remain in the infant for several months. Okay, so artificial would be like antiserums and antitoxins. Natural would be mother to fetus. Okay, artificial passive immunity. So tetanus is a disease caused by a bacteria that lives in soil, saliva, dust, and manure. The bacteria enters the body through a cut and has to be a pretty deep one. So tetanus kills quickly, so the body's natural primary response cannot occur. Therefore, people who have a deep cut and may have been infected with the tetanus bacteria will need to be given an injection of antitoxins. So these are given as a preparation of human antibodies. The antibodies are collected from donated blood from people who had recently been vaccinated against tetanus. So these antitoxins will provide an immediate response, but it is only temporary, right? Because the antibodies are not produced by your body's own cells. So the antibodies will be attacked as well by phagocytes in the liver and in the spleen once the body has enough time to launch that immune offense. Okay, so this is a table summarizing passive versus active immunity. Okay, so active immunity is where the antigen is encountered. There is a immune response. It can be several weeks during the primary response. Okay, there is a production of memory cells and the protection is permanent. Okay, passive immunity is where there isn't an antigen encountered. Okay, so this is like with the tetanus where you get a deep cut and you know that you need to protect yourself. So you go to the doctor, they give you a shot that has immune cells in there. Okay, so it's not your own immune response, but the time before the antibodies appear in the blood is immediate. Okay, so and there's no production of memory cells, and the protection is only temporary. Okay, so here's that. Here's a model showing immunity. It's either innate, like inborn genetic factors, or it's acquired. And if it's acquired, it's either active or passive. If it's active, it's natural, okay, like you've been through an exposure to an infectious agent, or it's artificial because you've had an immunization. Okay, if it's passive, you're either getting it naturally through maternal antibodies through breast milk or across the placenta, or it's artificial because you're receiving antibodies from other sources. All right, here is that model again. Okay, so vaccines are preparations containing antigenic material. That means it can contain a whole live microorganism, a dead one, a harmless form, which means it's been attenuated, a harmless form of a toxin like a toxoid, or a preparation of surface antigens. Vaccines can be given via an intravenous or an intramuscular injection, or some of them can be taken orally. Vaccines can be used to mimic an immune response that is caused from a natural infection. So when a vaccine contains living organisms, they reproduce slowly and the immune system will continuously be presented with a large dose of antigens. These vaccines are highly effective and may give a lifetime's protection. But if a vaccine is made from dead bacteria or viruses, it will not mimic a response caused by a natural infection and is somewhat less effective. So these vaccines need booster injections to simulate secondary responses that will then give that person enhanced protection. Okay, we're all familiar with vaccinations and booster shots. Okay, so problems with vaccines. Unfortunately, there are problems that have been associated with vaccines. Some people do not respond well at all to vaccinations. And it can be due to a defective immune system, which will then prevent the production of the necessary B and T cell clones. Also, it could be because the person suffers from malnutrition, right? Especially if you have protein energy malnutrition. It causes the person not to have enough proteins to make the antibodies that suddenly your body has been flooded with, okay, with the antigen. So these people run a high risk of developing the infectious diseases that they're being vaccinated against and passing them on to people with no immunity. Some people that are vaccinated with a live virus can pass it out in their feces during primary response, which can cause others to become infected. But this can be avoided by giving herd immunity. So this is when a large number of people, a high percentage of a particular population has been vaccinated at the same time. Okay, and herd immunity is very important because then if there are individuals, if there are few individuals who don't respond well to the vaccines, it's generally still safe because everyone else has been vaccinated and carry immunity. So therefore, the likelihood of any disease becoming endemic in that population is low. Okay, so antigenic variation is when a pathogen alters its surface protein so that it can invade a cell. And this can cause a problem because antibodies that are used to kill a previous strain are then not recognized. So if a minor change occurs to that antigen, which is called antigenic drift, the memory cells can still recognize it and start a secondary response. But if it's a major change, okay, which is called antigenic shift, a vaccination that was given will no longer be effective. Okay, and this commonly happens, this antigenic shift, in flu viruses. Okay, the WHO recommends the type of vaccine to be administered based on the antigens that are common. So this is why vaccines like the flu vaccine change every year. There is no vaccine for the common cold and no effective vaccines against diseases that are caused by protocysts. So the rhinovirus that causes most cold has at least 113 different strains. So that makes it really impossible to develop a vaccine that can protect a person against all of those strains. Protists are eukaryotes that have many more genes than bacteria and viruses. So they can have up to thousands of antigens on their cell surface. And some even have different antigens during different stages in their life cycle. So that means a vaccine is nearly impossible to make. Okay, Plasmodium passed through 3 stages within the human host, each with a different antigen. So in order for a vaccine to be effective, it must be specific to the infective stage and cause an effective response in the time between the mosquito bite and the infection of the liver cells. So Plasmodium, like with malaria, okay? And then trypsinoma, which is the agent that causes the sleeping sickness, has 1,000 different antigens that change every 4 to 5 cycles, making it absolutely impossible for the immune system to be effective. Okay. Another problem is that some pathogens evade an attack by the immune system by living inside cells. So parasitic worms can conceal themselves by covering their bodies in host proteins. This makes them invisible to the immune system, which will prevent them from being attacked. Other pathogens suppress the immune system by parasitizing cells like macrophages and T cells. Developing vaccines for these type of pathogens is extremely difficult because there is a short period of time for an immune response to occur before the pathogen ends up hiding. So Vibrio cholerae remains in the intestine where it is not capable of being reached by antibodies. The vaccine is injected, so it is unable to stimulate antibody production in the intestine. It also cannot stimulate the production of antitoxin. So, but if you take an oral vaccine for cholera, then it will get into your intestine. Okay, smallpox. Smallpox has an acute, was an acute, highly infectious disease caused by the variola virus that was transmitted by direct contact between people. So it causes red spots containing a transparent fluid all over your body. The eyelids become swollen, sometimes shut. If people recover from it, they would be permanently blind potentially and disfigured by scabs that were left when the pustules dried out. So between 12 to 30% of people, and I've heard estimates even up to 50% who have smallpox died. So the WHO began the smallpox eradication program in 1956. It was composed of vaccination and surveillance. So vaccinations of over 80% of populations that were at risk occurred. And so by performing ring vaccinations, everyone who may have come into contact with an infected person became protected. And in 1980, the WHO declared the world free of smallpox. So the eradication program was successful due to many factors. The virus was stable. It does not mutate a ton. So vaccines could be used everywhere and then also became cheap to produce. The vaccine was made from a harmless strain of a similar virus. So it was effective due to the fact that it was a live vaccine and it didn't mutate a lot. The vaccine was freeze-dried and could be kept at high temperatures for as long as 6 months, so it could travel around and be stored in doctors' offices. Infected people were easy to identify. The vaccine was easy to administer, and was even more effective after the development of the reusable needle. The smallpox virus did not linger in the body after an infection, and the virus did not infect animals, which made it really easy to break that cycle. And many 16 to 17-year-olds became really enthusiastic vaccinators and suppliers of information about cases because smallpox was such a big threat. There was this movement in the 50s of 16 and 17-year-old teenagers really like pushing their older and younger relatives to get vaccinated. This really It really became effective because it was coming from people who are most at risk. Okay, measles. Measles is caused by a virus that is spread by airborne droplets. And it causes a rash and a fever, and it potentially has fatal complications. However, it rarely affects infants under 8 months because they have that passive immunity from their mother. So it is a major disease of developing countries because of overcrowding and unsanitary conditions. It will easily infect malnourished infants that suffer from vitamin A deficiency. Measles has caused many cases of childhood blindness and severe brain damage. In 1993, there were an estimated 45 million cases of measles and 1.16 million deaths, making it the 9th leading cause of death worldwide. Measles is preventable and could be eradicated by a worldwide surveillance and vaccination program. However, a one-dose vaccination has not eliminated the disease due to a poor response to the vaccine by children. Some children need several boosters to develop immunity, but it is difficult to give boosters and to follow cases of measles and trace its contacts. Measles is very infectious, and 93 to 95% immunity, herd immunity, is needed to prevent the transmission. So the vaccine is about 95% effective, so then the whole population must be vaccinated, and infants must be vaccinated within 8 months of birth to prevent the transmission. So many countries have achieved 80% or more with just vaccination alone, but with the anti-vaccination movement that's been occurring, measles is starting to be a disease that's coming back, right? Because we need that 95% herd immunity. As people are not vaccinating their children, then herd immunity goes down. And that's the thing about herd immunity, is that especially with this type of disease where sometimes people need multiple boosters to retain immunity, you don't necessarily know if you need a booster. So let's say you've gotten the measles vaccine and you feel like you're safe, your parents think you're safe, you've gotten a booster, you don't really realize that your body is kind of rejecting that, or it needs to be refreshed. You need that booster. But you feel safe because you got the vaccinations. But someone, one of your classmates who hasn't gotten vaccinated, can spread that disease to you, even though you've been vaccinated. And that's why herd immunity is so important, because if that student next to you had gotten vaccinated, then the chance of you getting it, even though you need that booster, drastically decreases, right? So that's why vaccinations are so important. And they're really responsible for our livelihood and the quality of life that we enjoy. Okay, and on that note, I will say goodbye. Again, make sure you look over your textbook, look at all those pop-outs. They really like to ask about herd immunity on the Cambridge exam, and about how different countries have really had campaigns to get people vaccinated. So make sure you're reading all of that, and I'll see you in class.